Describe the mechanism of entry of carbon skeleton in amino acid metabolism. This carbon skeleton, which is coming from the degradation of amino acids, is not considered as the waste product. Okay, it is biologically significant. That's why we are discussing this carbon skeleton here. Okay, discuss the process of vitamin B12 cobalamin as a cofactor and methyl donor in metabolism of these amino acids. So these are the objectives of today's lecture. Okay, the carbon skeleton that is coming from the degradation or the catabolism of amino acids, it has different fates depending upon the condition, depending upon the requirement of the body. The amino acids catabolism releases amino group either from transamination reactions or from deamination reactions. The amino group which is released from the amino acid is transferred towards the liver to join urea cycle, and it converts into urea to be excreted out of the body, because this amino group is considered as the waste product of amino acid catabolism. After removing or after releasing the amino group, this amino acid becomes, first it becomes keto acid, then it becomes any of the intermediate of different metabolic pathways. So the amino acid structure catabolized into two major metabolites. The first one is the amino group, which is converted into the urea, and the second one is, after the removal of the amino group, the rest of the structure of the amino acid is called as carbon skeleton. So the remaining structure after losing the amino group from the amino acid is called as the carbon skeleton of amino acids. The catabolism of carbon skeleton of amino acids is either one or two of the following intermediates. There are total 7 intermediates that are coming from the catabolism of the amino acids. These are the carbon skeleton of the amino acids, and they are also involved in the metabolic pathways of some other biomolecules. These carbon skeletons are not only associated with the amino acids. For example, the first one is the oxaloacetate, which is coming from the degradation of some amino acids, as well as this oxaloacetate is involved in Krebs cycle, TCA cycle. The first step of Krebs cycle is utilization of oxaloacetate. This oxaloacetate can also convert into glucose by the process of gluconeogenesis. The second one is the alpha ketoglutarate, again an intermediate of TCA cycle, Krebs cycle. Third one is the pyruvate, again an intermediate of glucose metabolism. This pyruvate can enter into the TCA cycle, or this pyruvate can enter into gluconeogenesis for synthesis of glucose, depending upon the requirement of the body. Fumarate, another intermediate of TCA cycle. Succinyl-CoA, again TCA cycle intermediate. Sixth one is the acetyl-CoA. This acetyl-CoA can enter into the TCA cycle, but this acetyl-CoA can also be utilized for the synthesis of fatty acids. And the last one, the 7th intermediate, which can be obtained from the degradation or the catabolism of amino acid, is acetoacetate, a 4-carbon compound that contains a ketone group. Out of these 7, the last 2, acetyl-CoA and acetoacetate, they are associated with the fatty acid metabolism. They contain a ketone group. So they are called as ketone bodies. And the above 5 intermediates or the above 5 carbon skeletons that are coming from the amino acid degradation, they are largely associated with carbohydrate metabolism. They can give energy when enters into the TCA cycle, or some of these intermediates can give glucose by the process of gluconeogenesis. So the amino acid catabolism can give the intermediates of carbohydrate metabolism, or it can give the intermediates that can join fatty acid metabolism. Depending upon the end product, there are different types of the amino acids. These amino acids are not only the building blocks of proteins and peptides. As we all know, the major function, the primary and the basic function of the amino acids is the synthesis of protein, and they are said that they are the building blocks of proteins as well as some short peptides and polypeptides. But these amino acids can also synthesize glucose by the process of gluconeogenesis. These amino acids can also synthesize fatty acids if they give acetyl-CoA or acetoacetate. These amino acids can also synthesize some other specialized products. Most of the nitrogen containing compounds that are present in our body, they are synthesized from the amino acids. End products of glucogenic amino acids can oxidize to carbon dioxide and water through TCA cycle and yield little amount of energy. The basic function of proteins and amino acids is not to give energy as does by carbohydrate, glucose and fats. When the body is deficient in glucose and fatty acids, then the proteins and the amino acid catabolism joins TCA cycle, or it joins catabolic pathway that gives energy. Under normal circumstances, the amino acid catabolism or the protein catabolism does not indicate energy production. So amino acid gives energy only when the body is deficient in glucose. So these amino acids convert themselves into glucose through gluconeogenesis. Then this glucose gives energy through glycolysis and TCA cycle. So depending upon the end product, as I said, the initial 5 intermediates are not only the carbon skeleton coming from amino acid catabolism, but they are also the intermediate of carbohydrate metabolism. And the last 2, acetyl-CoA and acetoacetate, are the metabolic end products of the amino acid catabolism, as well as they can join fatty acid metabolism. So we have different types of the amino acids, like glucogenic amino acids, when the end product of amino acid degradation is pyruvate or TCA cycle intermediate that can enter into the gluconeogenesis. The amino acid could be a ketogenic amino acid when the end product of amino acid catabolism is acetyl-CoA or acetoacetate, then it is said to be a ketogenic amino acid. And few amino acids has dual nature. They give 2 types of the end products. The first one is the glucogenic and the second one could be acetyl-CoA or acetoacetate. So it is glucogenic as well as ketogenic amino acid. When the amino acid degradation yields intermediates of carbohydrate metabolism as well as fatty acid metabolism. So in the right hand side, you have a table that contains examples, the names of the amino acids that are glucogenic, the amino acids that are gluco and ketogenic both, and the amino acids that are ketogenic. So we have total 13 amino acids. Out of these 13 amino acids, the 9 amino acids are non-essential and the 4 amino acids are the essential amino acids, in which the arginine, is a semi-essential amino acid. This arginine is sometimes placed in essential category or sometimes it is placed in non-essential category. Arginine is a semi-essential amino acid and rest of the three, histidine, methionine and valine, are the essential amino acids. Their catabolic end products are the intermediates of carbohydrate metabolism. They can join TCA cycle or they can give rise to glucose. So they are glucogenic amino acids. We have total 5 types of the amino acids that are glucogenic and ketogenic both. Tyrosine, non-essential amino acid, but it is coming from degradation of phenylalanine. And the 4 essential amino acids, isoleucine, phenylalanine, tryptophan and threonine. They are glucogenic as well as ketogenic. We have total 2 amino acids and they both are the essential amino acids, leucine and lysine. They are ketogenic amino acids because their end product is acetyl-CoA and acetoacetate. So we have total 20 amino acids and out of these 20, 13 are glucogenic, 2 are ketogenic and 5 are glucogenic, ketogenic both. You will have to learn the names of amino acids in each category, but just to make it easy, just only learn the name of the ketogenic amino acids and gluco-keto both amino acids. The rest of the 13 amino acids could be included in the glucogenic category. To make it easy, just learn the ketogenic amino acids. Only 2, leucine and lysine. Gluco and keto both. Tyrosine, isoleucine, phenylalanine, tryptophan, threonine. And rest of the amino acids could be included in the glucogenic category. So most of the amino acids are glucogenic. So they can synthesize glucose through gluconeogenesis. So this is the significance of carbon skeleton that is coming from the degradation of amino acids. This carbon skeleton is not considered as the waste product as it is coming from the catabolic pathway of the amino acids, but it is not the waste product because this carbon skeleton has a worth. It can enters into gluconeogenesis, it can enters into TCA cycle, it can enters into fatty acid synthesis. So again, we have categorized different amino acids. The red color amino acid shows the glucogenic amino acids. The name of the amino acids that are written in green color, they are ketogenic amino acids. And the amino acids that are written in the purple color, they are glucogenic and ketogenic both in nature. Okay. So here you can see the end product of alanine, cysteine, glycine, serine, threonine and tryptophan is pyruvate. But this pyruvate can convert into acetyl-CoA to join TCA cycle. And this pyruvate can be converted into oxaloacetate, and this oxaloacetate through phosphoenol pyruvate synthesize glucose by gluconeogenesis. Acetyl-CoA, which is coming from the degradation of tryptophan, tyrosine, phenylalanine, threonine, isoleucine, lysine and leucine. If oxaloacetate is available, this acetyl-CoA combines with oxaloacetate and enters into the citrate TCA cycle. But if oxaloacetate is not available, this acetyl-CoA can be utilized for fatty acid synthesis or for ketone bodies formation. Alpha ketoglutarate is coming from the degradation of glutamate, arginine, proline, histidine and glutamine. This alpha ketoglutarate is an intermediate of TCA cycle. Succinyl-CoA or succinate. This succinyl-CoA is coming from degradation of threonine, methionine, isoleucine and valine. Fumarate is coming from the degradation of tyrosine, phenylalanine and aspartate. And again, oxaloacetate is coming from the degradation of aspartate and asparagine. So this is the significance of carbon skeleton that is coming from the degradation of amino acids. This carbon skeleton can enters into the TCA cycle, can enters into the gluconeogenesis, or if the carbon skeleton is acetyl-CoA or acetoacetyl-CoA, then it can be utilized for fatty acid synthesis or for the formation of ketone bodies. The formation of ketone bodies is largely associated with fatty acid metabolism. So when you will discuss about the fatty acid metabolism, then there will be a detailed lecture about formation of ketone bodies. The carbon skeleton can be entered into amino acid metabolism. Non-essential amino acids that are synthesized in our body, they are synthesized by transamination or amination of carbon skeleton. If the carbon skeleton is aminated either through amination or through transamination, this carbon skeleton can become an amino acid. So the synthesis of non-essential amino acids can occur into our body when an amino group is transferred from an amino acid through transamination or from any of the source to a carbon skeleton, then this carbon skeleton becomes the amino acid. So the amino group donor for the synthesis of amino acid is usually glutamate if it occurs through transamination reactions. Like alpha ketoglutarate is the acceptor of amino group during transamination. Similarly, glutamate is the donor of amino group for transamination. And sometimes the amino group as free ammonia-amines can be entered into the carbon skeleton for the synthesis of amino acids. The carbon skeleton that are required for the synthesis of amino acids, they are the intermediates of carbohydrate metabolism and fat metabolism. This is the summary of synthesis of some amino acids from carbon skeleton. We have total 20 type of the amino acids. Out of these 20, our body can synthesize 11 amino acids. The 11th arginine is the semi-essential, but we can synthesize arginine. Usually arginine is essential amino acids for infants and children. But for adults, this arginine becomes non-essential because of active urea cycle, and our urea cycle also utilizes arginine. Okay, so we have total 20 types of the amino acids. Out of these 20, we can synthesize 11 amino acids. 9 are the essential amino acids, but these essential amino acids are not synthesized in our body, but can be synthesized by some other organisms like bacteria, like some plants, because the source of the essential amino acids are usually another source other than humans. So the oxaloacetate, which is the intermediate of TCA cycle, this oxaloacetate can synthesize aspartate, and from aspartate, asparagine, methionine, threonine, and lysine is synthesized. And threonine can synthesize isoleucine. So oxaloacetate is the carbon skeleton for the synthesis of aspartate, asparagine, methionine, threonine, lysine, and isoleucine. Pyruvate, which is coming from the glycolysis of glucose. This pyruvate can synthesize alanine, valine, and leucine, either through transamination or through amination. Ribose 5-phosphate, a 5-carbon sugar. It gives histidine. Phosphoenol pyruvate, a derivative of pyruvate, when combined with erythrose 4-phosphate, a 4-carbon sugar, It gives aromatic amino acids phenylalanine, tyrosine, and tryptophan. And this phenylalanine in humans can form tyrosine. So the tyrosine is synthesized from phenylalanine for humans, but this tyrosine can directly be synthesized in other organisms when phosphoenol pyruvate combines with erythrose 4-phosphate. Erythrose 4-phosphate is a 4-carbon sugar. Alpha ketoglutarate, a TCA cycle intermediate, can form glutamate, and this glutamate can synthesize glutamine, proline, and arginine. 3-phosphoglycerate, a derivative of 3-carbon sugar glyceraldehyde. 3-phosphoglycerate, it can synthesize amino acid serine, and from serine we obtain glycine and cysteine. If you remember, cysteine, which is a sulfur containing amino acid, it is synthesized from serine and methionine. Serine is the carbon skeleton for cysteine, and methionine is the donor of sulfur in the form of homocysteine. The yellow color amino acids in this diagram shows the amino acids that can synthesize another amino acid, like aspartate can synthesize asparagine, methionine, threonine, and lysine. And the threonine is yellow highlighted because threonine can give rise isoleucine. So the yellow highlighted amino acid shows that they are the precursor for another amino acid. And the few amino acids are in bold font, like methionine, threonine, isoleucine, lysine, valine, leucine, histidine, tryptophan, phenylalanine. These all are essential amino acids for humans. The yellow highlighted amino acid means they can synthesize another amino acids, and the amino acids that are in bold font, it means they are essential amino acids. So human body can synthesize only non-essential amino acids, and essential amino acids cannot synthesize in our body, but plants and bacteria and some other organisms can synthesize also these essential amino acids. So why the human body cannot synthesize essential amino acids? We have total 20 type of the amino acids. Out of these 20, 9 are the essential amino acids. Here, this is a graph that shows the number of steps involved in the synthesis of different amino acids. We have total 4 amino acids that are synthesized only in one step. We have total 2 amino acids that are synthesized in 2 steps. We have an amino acid that is synthesized only in 3 steps. 2 amino acids are synthesized in the 4 steps. The blue color shows the amino acids that are non-essential, that are synthesized by the human body. And the pink color shows the amino acids that are not synthesized in our body. So here you can see the pink highlighted amino acids 1, 2, 3, 4, 5, 6, 7, 8, 9. We have total 9 highlighted pink color amino acids that involves multiple steps for their synthesis. Like we have at number 10, you can see 2 amino acids. The first one is blue highlighted and the second one is pink highlighted. And at number 16, there is an amino acid, a single amino acid that is synthesized in 16 steps. One amino acid is synthesized in 12 steps. One amino acid in 11 steps. So when the synthesis of amino acid involves multiple steps, it is thought that during evolution of human beings, there are some genetic fault, there are some mutation occurs in the human body. So because of these mutation, the enzymes of the amino acid synthetic pathway that are involved in the synthesis of amino acids of more than 8 steps or more than 5, 6 steps, that becomes deficient during evolution. So the human body now becomes deficient in few amino acids. So we have some essential amino acids that must be obtained from the diet. Okay. So this is the reason why few amino acids are not synthesized by the human body, but plants can synthesize. Role of B12 in amino acid metabolism. B12 cobalamin is required for the activity of the enzyme methylmalonyl-CoA mutase that converts L-methylmalonyl-CoA to succinyl-CoA. This methylmalonyl-CoA is coming from the degradation of different amino acids, and it finally converts into succinyl-CoA by the enzyme methylmalonyl-CoA mutase. This mutase enzymes, when convert methylmalonyl-CoA into succinyl-CoA, it requires cobalamin as a prosthetic group. This cobalamin B12 is also required for the activity of enzyme methionine synthase, which is involved in the synthesis of methionine from homocysteine. When homocysteine becomes methylated, the donor of methyl group is B12 and B9 tetrahydrofolate. This reaction has been discussed when we are discussing about the metabolism of sulfur amino acids. So these two enzymes requires cobalamin. Methionine synthase also utilizes B9 folate along with B12. Tetrahydrofolate or folate, which is the donor of methyl group. In different biochemical reactions, we have different types of the reactions that involve the transfer of one carbon to another, from one carbon from a molecule to the another molecule. This one carbon contains, if 3 hydrogen, it becomes a methyl group. The single carbon along with 3 hydrogen, it becomes a methyl group. In different methylation reactions, the donor of methyl group is tetrahydrofolate, or it could be S-adenosyl methionine, SAM, the active form of methionine, and the tetrahydrofolate, the active form of folic acid. If the carbon group is donated in the form of methylene group CH2 or methynyl group, carbon along with one hydrogen, hydrogen and oxygen, HNH, single carbon along with hydrogen or oxygen or nitrogen, then the donor of methylene and methynyl group is tetrahydrofolate. There are different biochemical reactions occur in the body that utilizes the transfer of carbon along with some hydrogens. If this carbon is in the form of methyl group, then it is transferred from, usually it is transferred from tetrahydrofolate or S-adenosyl methionine. If this carbon is in the form of methylene group or methynyl group, then the donor of this carbon is tetrahydrofolate. So tetrahydrofolate exists in different states, in different form. It can donate a methyl group, it can donate a methylene group, and it can donate a methynyl group. Sometimes these reactions also involve the transfer of carbon in the form of carbon dioxide in different carboxylation reactions. When the carbon is transferred in the form of carbon dioxide instead of a hydrocarbon like methyl, methynyl, methylene, this carbon in form of carbon dioxide during different carboxylation reactions is transferred in the presence of biotin, B7. So tetrahydrofolate B9 is donor of CH3, CH2 or CH in different reactions, and biotin act as a prosthetic group of different enzymes that are involved in carboxylation reactions by the addition of carbon dioxide instead of carbon with hydrogen. So this is the role of vitamin B12, cobalamin in amino acid metabolism. On the right hand side, you can see methylmalonyl-CoA, which is coming from the degradation of methionine, valine, isoleucine, and threonine. And this methylmalonyl CoA is also coming from the degradation of some fatty acids which are odd chain, like propionic acid. This odd chain fatty acids also gives methylmalonyl CoA. This methylmalonyl CoA converts into succinyl CoA, and the succinyl CoA can enters into the TCA cycle, or it can enters into the biosynthesis of heme. When succinyl CoA combines with glycine, the heme biosynthesis pathway is initiated. So this methylmalonyl CoA mutase enzyme requires vitamin B12 in its adenosyl form, adenosylcobalamin. This vitamin B12 is also required for the activity of enzyme methionine synthase. This homocysteine, upon methylation, becomes methionine by the enzyme methionine synthase. It requires vitamin B12 as a donor of methyl group, and this methyl group is coming from tetrahydrofolate. So this tetrahydrofolate gives methyl group to B12, and this B12 acts as a methyl group donor. So there is a transfer of methyl group between three molecules as adenosylmethionine, tetrahydrofolate, and B12. And finally, this methyl group reached at homocysteine to synthesize methionine. This homocysteine can give rise to cysteine and propionyl CoA upon catabolism. PLP is vitamin B6 pyridoxal phosphate, the major and the principal prosthetic group for amino acid catabolic reactions. So pyridoxal phosphate is usually involved in transamination reactions. So this is the involvement of B12 in amino acid metabolism. Some specialized products of amino acids other than proteins. Glutathione, which is a tripeptide of glutamate, cysteine, and glycine. This glutathione has antioxidant activity. It is present in RBCs. It prevents our body from oxidative damage from some reactive oxygen species. The free radicals that are synthesized as a side byproduct of different oxidation reduction reactions, these free radicals can damage biomolecules. So naturally, our body has a natural system to neutralize the effect of these free radicals. The glutathione is one of the molecules that prevents our body from the harmful effects of free radicals, and these free radicals are highly reactive molecules that are coming from the different oxidative reduction reactions. Anserine and carnosine, these are the two dipeptides. Anserine is composed of beta-alanine and L-histidine, and beta-alanine is the amino acid alanine derivative that contains amino group at beta carbon instead of alpha carbon. Carnosine is beta-alanine in methylated form and L-histidine. So anserine and carnosine similar in structure somehow, but the only difference is the methylation. They both are present in muscles and brain. In muscles, they are involved in buffering mechanism of blood and antioxidant activity in muscles. And in brain, these anserine and carnosine has neuroprotective effects. Some other derivatives are some peptide hormones that are not the actual protein. They are short peptides like oxytocin from posterior pituitary. It composed of 9 amino acids. Vasopressin composed of 9 amino acids. They both are secreted from the posterior pituitary gland. Oxytocin is involved in contraction of smooth muscles. Vasopressin is involved in the reabsorption of water, glucagon is secreted from the pancreas, 29 amino acids, angiotensin, 10 amino acids, somatostatin, 14 amino acids. These are the some short peptides that are hormone in nature. Amino acids are the derivatives of some other nitrogen containing compounds. For example, heme. You have discussed about the synthetic pathway of heme. The first step of heme biosynthesis is the condensation of glycine with succinyl CoA that forms amino-levulinic acid, ALA, by the enzyme ALA synthase. Amino-levulinic acid then forms a pyrrole ring, then a tetrapyrrole, linear tetrapyrrole, cyclization, porphobilinogen, uroporphyrinogen, coproporphobilinogen, then heme. So the first step of heme biosynthesis, which is the regulatory step, requires amino acid glycine as a donor of nitrogen in porphyrin ring. So in the porphyrin ring, the nitrogen is coming from the glycine and the carbon skeleton is coming from the succinyl CoA. Heme is the prosthetic group of some other proteins like hemoglobin, myoglobin, cytochromes, tryptophan pyrrolase. Thyroid hormones T3 and T4, they are also synthesized by the amino acid tyrosine. So tyrosine is the precursor for the synthesis of T3 and T4 thyroid hormones from thyroid gland. Our thyroid glands contain a protein globin. The tyrosine residue, when iodinated, the addition of iodine to this tyrosine residue of globin protein makes T3 and T4. If 3 iodines are added, it becomes T3, and if 4 iodines are iodinated on tyrosine structure, it becomes T4. So upon iodination of tyrosine, the tyrosine becomes thyroid hormone T3 or T4. Tyrosine is also the derivative of some catecholamine structures like dopamine. Dopamine is a neurotransmitter synthesized in the brain from hydroxylation and decarboxylation of tyrosine. Dopamine is a neurotransmitter which is involved in locomotor activity and mood regulation. This dopamine is synthesized from tyrosine. Norepinephrine and epinephrine, these are the hormones secreted from adrenal medulla, and they are also a neurotransmitter when they are synthesized in the brain. Norepinephrine and epinephrine are active during stress responses. They are also synthesized from tyrosine. Tyrosine gives dopamine upon hydroxylation and decarboxylation. Then this dopamine upon hydroxylation becomes norepinephrine, and norepinephrine upon methylation becomes epinephrine. So when epinephrine is synthesized from norepinephrine, there is a methylation reaction which is done in the presence of S-adenosylmethionine. Just to recall, when I was talking about the biological significance of SAM, S-adenosylmethionine, I give you the example of epinephrine. The epinephrine is also synthesized from the methylation by S-adenosylmethionine. Melanin is synthesized from the tyrosine. When you discuss about the aromatic amino acids catabolism, you have learned about the melanin. The deficiency of melanin leads to albinism. Serotonin, another neurotransmitter, when it is synthesized in the brain, the serotonin is also synthesized outside the brain, have large effect on GI system, digestive system पे इसका large effect होता है. The serotonin also have a vasoconstriction effect when it is present in blood vessels, and the serotonin controls our mood, our wake sleep cycle, our eating behaviors when it is synthesized from the brain. So the serotonin is synthesized from the essential amino acid tryptophan. Deficiency of tryptophan leads to deficiency of serotonin, and serotonin is involved in the regulation of mood. So deficiency of serotonin usually characterized by depression. Melatonin, a hormone which has sleep-inducing effect. This melatonin is also synthesized from the amino acid tryptophan. When tryptophan synthesized serotonin, then upon methylation by S-adenosylmethionine, this serotonin becomes melatonin. Melatonin is synthesized in the pineal gland in our brain. It can also synthesized in some peripheral tissues, but large production occurs in pineal gland in the brain. When serotonin is methylated by S-adenosylmethionine, it becomes melatonin. Gamma aminobutyric acid, a neurotransmitter that controls the release of other neurotransmitters. It is synthesized from the decarboxylation of glutamate. Histamine, that is active in allergy and inflammation responses. It is synthesized from the histidine. Histamine that is active during allergy and inflammation. This histamine is synthesized from the decarboxylation of histidine. Bile acids also utilized some amino acids like glycine, cysteine, or taurine. Carnitine, it is a nitrogen containing compound which is coming from the amino acid lysine and methionine. This carnitine is required for fatty acid oxidation. Carnitine is required. It is a carrier molecule, a transport molecule during fatty acid oxidation. Creatine, it is synthesized by the amino acid arginine, glycine, and S-adenosylmethionine as a donor of methyl group. This creatine is synthesized in the liver. It is present in the form of creatine phosphate in muscles, and it is required for skeletal muscles activity. Creatine, the energy molecule in the form of creatine phosphate present in skeletal muscles. The amino acids arginine can also synthesize nitric oxide. This is a short-lived active molecule for signal transduction. Nitric oxide by the enzyme nitric oxide synthase is synthesized from the arginine. So arginine can synthesize nitric oxide by the enzyme nitric oxide synthase, and this nitric oxide is a short-lived signal molecule involved in signal transduction processes. Besides all these derivatives, amino acids are also required for the synthesis of purines and pyrimidines nitrogen bases, adenine, guanine, cytosine, uracil, and thymine. The nitrogen base in these nitrogenous bases is also coming from the amino acids. So amino acids are also required for the synthesis of purines and pyrimidine bases for DNA and RNA synthesis. Okay, so you can read this from Harper's Biochemistry.